Welcome to Department of Botany, Kalna College. Myself, Dr. Somnath Mondol, and today's topic is population and evolutionary genetics. We will study these topics in multiple parts. In part 1, we will study about the Hardy-Weinberg law and the way to calculate allele frequency or gene frequency and genotypic frequencies in a population. So let's start. In mid-19th century, Alfred Russel Wallace and Charles Darwin identified natural selection as the mechanism of evolution. In his book in 1859 on the origin of species, Darwin presented comprehensive evidence that population and species are not fixed. Rather, they change, they evolve over time as a result of natural selection. In addition to natural selection, other forces like mutation, migration, genetic drift, all these factors individually or collectively can cause a change in allele frequency and bring about evolutionary divergence that eventually may result in speciation, that is, the formation of new species. Populations that are reproductively isolated are regarded as separate species. Genetic changes within populations can modify a species over time and transform it into another species, or may cause the species to split into two or more species. Population geneticists investigate the patterns of genetic variations within and among groups of interbreeding individuals. And thus, population genetics has become an integral part, a subdivision of evolutionary biology. A population can be defined as a group of individuals belonging to the same species that live in a defined geographic area and actually or potentially interbreed. The genetic information carried by members of a population constitutes the population's gene pool, or if I say in other way, that total number of genes in haploid set of chromosomes in a population is known as gene pool of that population. Microevolution can be defined as the evolutionary change within populations of a species, but macroevolution is the evolutionary event leading to emergence of a new species or other taxonomic groups. Change in gene frequency in a population denotes whether a population is undergoing evolution or not. Now, how to calculate gene frequency? Total number of genes at a locus divided by total number of genes in the same locus in a population. Thus, we can calculate gene frequency. We will discuss a number of examples to explain this. Now, the relationship between relative proportions of allele in the gene pool and the frequencies of different genotypes in a population was described by the British mathematician Hardy and the German physician Wilhelm Weinberg. This model, called the Hardy-Weinberg law, describes what happens to alleles and genotypes in an ideal population. So what is that so-called ideal population? That is infinitely large with random matings, and the population that is not subject to any evolutionary forces like mutation, migration, or selection. Under these conditions, Hardy and Weinberg model makes two predictions. Number one, the frequencies of alleles in the gene pool do not change over time. And number two, if two alleles at a locus, for example, is capital A and small a, then as we get an equation of p square plus 2pq plus q square is equal to 1, where p square is the genotype capital A, that is the homozygous dominant. Q square is the genotype small a, that is the homozygous recessive, and 2pq is the heterozygous genotype, that is capital A, small a. Therefore, the dominant or wild type allele capital A, the frequency of capital A is P, and the frequency of the mutated recessive allele small a is Q. Now, the assumptions about the theoretical population discovered by Hardy and Weinberg law. First, individuals of all genotypes have equal rates of survival and equal reproductive success. That is, there is no selection pressure. That is quite unlikely in a real population. No new alleles are created or converted from one allele to another by mutation. In reality, there are so many mutagenic agents. Therefore, one allele, a dominant allele, can be converted into a recessive one. Individuals do not migrate into or out of the population. Yes, migration or migrating population is a characteristic pattern. In birds, we can see migration. Now, in terms of corona, we can see there is migration, lots of migrating peoples. The population is infinitely large, which in practical terms means that the population is large enough that sampling errors can be negligible, and individuals in the population mate randomly. Now, the Hardy-Weinberg law can be applied in human populations. Let's have some examples. First, of calculating allele frequency. Case study 1. The CCR5 gene, that means the CC chemokine receptor 5 protein present on the cell surface is a receptor of HIV-1. Now, CCR5 gene, the wild type allele is called or referred as 1, and the mutated allele is referred as del32. Why del32? Because the mutated gene has a 32 base pair deletion mutation. That is why del32. HIV-1 can recognize the wild type receptor, but it cannot recognize the mutated receptor or del32 receptor. Thus, people with homozygous dominant genotype, that is one allele at both the loci, will be affected, subjected to HIV-1 infection. However, people with del32 genotype, that is homozygous recessive genotype, will be safe because the HIV-1 will not recognize the mutated receptor protein. But the heterozygous 1 by del32, that is having one or wild type allele at one locus and the mutated locus on the mutated allele on the other locus, will be susceptible to HIV-1, but the disease progression will be slow because 50% of the receptors will be mutated, not recognizable by the HIV-1. Therefore, suppose in a population, the genotypes, the homozygous dominant genotype frequency is 79, the heterozygous genotype frequency is 20, and the homozygous recessive phenotype genotype frequency is 1. Therefore, what is or how we can calculate the allele frequency from this data? There are two methods. First of all, counting allele method. We will draw a table like that, and in this table we will calculate the number of 1 alleles and number of del 32 alleles in that population. Now, for example, there are 79 homozygous dominant people. Therefore, on both loci of 79 people, there are 1 allele. Therefore, 79 into 2, 158 alleles. In case of heterozygous genotype, that is 1 allele is present only in 1 locus. Therefore, 20 into 1, 20 number of 1 allele present in the heterozygous population. And in case of the homozygous recessive population, there will be no 1 allele. Thus, the total number of 1 allele is 178. Similarly, we can calculate the total numbers of del 32 alleles in heterozygous and homozygous recessive population, and this is 22. Therefore, 178 plus 22 is equal to 200 alleles. Now, out of 200 alleles, there are 178 1 allele and 22 del 32 allele. Therefore, their frequency is respectively divided by 200 each is 89% and 11% respectively. So in this way, by counting alleles, we can calculate the allele frequency of certain genes in a population. In the second method, that is called the from genotype frequency to the allele frequency. Now, how we can calculate? So the genotype frequencies, we know that 79 homozygous dominant, 20 heterozygous, and 1 homozygous recessive. Therefore, total is 100. Total 100 individuals has been surveyed. Now, out of 179, that is 0.79 is the genotype frequency of homozygous dominant. Out of 120, that is 0.20 is the genotypic frequency of heterozygous, and 1 out of 100, that is 0.01 is the genotypic frequency of del 32, that is the homozygous recessive genotype. Now, therefore, from genotype to allele frequency, let's calculate. The allele frequency of 1 allele should be 0.79, where both loci has 1 allele, plus half into 0.20. Why half? Because 0.20 is the heterozygous, where half is 1 allele, half is del 32 allele. That is why half into 0.20. So the value is again 89%. In the same way, we can calculate the frequency of del 32 allele in the sample. That is half into 0.20 plus 0.01 equal to 11%. So the data is the same like before. So the two methods are the contrasting ones. Now, case study 2. Suppose, based on the same CCR5 gene, the genotype and the frequencies are given. And the question is, so whether these two alleles violate Hardy-Weinberg equilibrium or not. We will make a similar chart of counting allele method to calculate the allele frequency, as there are 223 homozygous dominant genotypes, there will be 223 into 2, 446 1 allele, whereas 57 heterozygous, therefore 57 into 1 equal to 57 1 allele, in heterozygous. In homozygous recessive, there will be no one allele. Therefore, 446 plus 557 equal to 503. So total 503 are one allele. Similarly, del 32 alleles will be calculated. In case of homozygous dominant, there will be no del 32 allele. In heterozygous, there will be 57 into 1, 57 heterozygous allele. In case of homozygous recessive, there will be 3 into 2 equal to 6. Why into 2? Because on both loci there are del 32. That is why 3 into 2, 6. That is 57 plus 6 equal to 63 del 32 allele. Therefore, 503 plus 63 equal to 566 total number of alleles. So allele frequency 503 divided by 566 equal to 89% one allele, and del 32 allele frequency 63 by 566 equal to 11%. So the allele frequency in that population has been calculated. Now the expected genotype frequencies will be, so here according to Hardy-Weinberg, P is equal to the one allele. Therefore P square is equal to the homozygous dominant genotype, that is 0.89 whole square equal to 0.792. Accordingly Q is equal to the del 32 allele. Therefore Q square is equal to del 32, that is the homozygous recessive genotype. Therefore 0.11 whole square equal to 0.012, and the 2PQ, that is the heterozygous genotype, 1 by del 32, this will be 2 into P into Q, that is 0.196. So these are the expected genotypic frequencies. Now what are the observed genotypic frequencies? Let's calculate like before. Genotype frequency. So 223 out of 283 are wild type. Therefore frequency is 223 divided by 283 equal to 0.788. Similarly, heterozygous and homozygous recessive frequencies were also calculated, that is 0.201 and 0.011 respectively. Therefore, now we can compare between our expected data and observed data. Expected data and observed data, expected homozygous dominant frequency is 0.792 and the observed is 0.788. Similarly, expected heterozygous genotype frequency is 0.196, observed 0.201, and that of homozygous recessive expected is 0.012 and observed 0.011. Therefore, they are quite close enough. Now, these expected frequencies are nearly identical, but still we will calculate the chi-square value that we have already studied how to find a chi-square value, and this value here will be very tiny, 0.00023. Now in 5% probability that the chi-square value will be provided, or the t table will be provided, from where we can see that the chi-square value at probability 5% probability is 3.84, and here our data is very low, that is 0.00023. so therefore our result is a significant one. So therefore these two alleles in that sample human population are not violating Hardy-Weinberg equilibrium. In case study 3, we will calculate multiple allele frequency using the Hardy-Weinberg law. So ABO blood group system is a well-known multiple allele system where IA, IB and IO, these three alleles are there. So here there will be P plus Q plus R is equal to 1. where P is equal to IA, Q is equal to IB, and R equal to IO allele. Therefore, P plus Q plus R whole square is equal to 1. Now, this data has been provided that phenotype A blood group, frequency is 0.53, B blood group, frequency 0.12, AB blood group, frequency is 0.08, and O blood group, frequency is 0.26. You need to get the genotype frequencies. Now, we have to calculate that, and we have to take care that A blood group means the genotype can be IA-IA or IA-IO. Similarly, B blood group means the genotype can be IB-IB or IB-IO. Similarly, AB blood group means both the alleles are present, IA-IB, and O blood group means IO-IO. Therefore, here if A equal to P, B equal to Q, and O blood group is equal to I or IO is equal to R, then P square is IA-IA, Q square is equal to IB-IB genotype, and R square is equal to IO-IO genotype. Therefore, IA-IO is equal to 2PR, IB-IO is equal to 2QR, and IA-IB is equal to 2PQ. Now, after detecting these, we will go through the calculations. Therefore, O blood group is equal to R square, whose phenotypic frequency is 0.26 given. Therefore, R equal to square root of 0.26 is equal to 0.51. Now, A blood group means P square plus 2PR, and the phenotypic frequency is given 0.53. Therefore, P square plus 2PR is equal to 0.53. If we add R square on both sides, that is P square plus 2PR plus R square is equal to 0.53 plus 0.26, that is the value of R square, is equal to 0.79. So here comes the formula, P plus R whole square is equal to 0.79. Therefore, P plus R is equal to square root of 0.79, that is 0.89. Now we know the value of R, that is 0.51. So by subtracting 0.51 from 0.89, we get the value of P, that is 0.38. Now we get the value of P, we get the value of R. Therefore, calculating the value of Q will be the easiest. As we know, P plus Q plus R is equal to 1. Therefore, by subtracting those two values from 1, we get Q is equal to 0.11. Therefore, we get the values of P, Q and R. So now genotypic frequencies, the calculation will be a cakewalk. So the same chart where IA is equal to P square, therefore square the value of P. So this is the genotype frequency of IA, square the value of B, that is 0.11, that is will be the genotypic frequency of B blood group, IB. Now IA-IO, that is 2PR, therefore 2 into P into R, multiply the values 0.39, that is the heterozygous A blood group genotype frequency. Similarly, heterozygous B blood group, AB blood group and O blood group frequencies can be calculated, as we know the values of P, Q and R. So in this way, in a population, we can calculate the frequencies of multiple alleles as well as their genotypic frequencies. Now, case study 3, calculating allele frequencies for X-linked traits. Red-green color blindness is a well-known X-linked mutation. And suppose in a population 8% human males are colorblind. Now you need to calculate the frequency of carrier females in that population. So how should we progress? Therefore, the frequency of color blindness is 8%, 8 out of 100, that is 0.08. In other words, 8% of X chromosomes carry the mutated allele. In other 92% X chromosomes, thereby the dominant allele is present. Let P is equal to the normal dominant allele and Q is equal to the colorblind allele. Now, the value of P is equal to therefore 0.92, 92%, and the value of Q is equal to 0.08, that is 8%. Therefore, the frequency of colorblind females will be, so females, if females need to be colorblind, both the X chromosomes need to have the colorblind allele or the recessive allele, that is Q, or Q square is going to be the colorblind female. So if colorblind female frequency is what you want, that is going to be Q square, or 0.08 whole square is equal to 0.0064. But here the question is, what is the frequency of carrier female? So carrier female is going to be 2pq. As we know, 2pq is the heterozygous genotype. Therefore, we know the value of P, we know the value of Q. So 2pq is equal to 2 into P into Q, that is 0.147. Therefore, 14.7% females will carry the allele for color blindness and can pass it to their children, although they will have a normal color vision. So that's how we can calculate allele frequencies, the genotypic frequencies in a population by using the Hardy-Weinberg law. That's all for today. Thank you.